Hello, everyone, and welcome to today's webinar, Cell Dive, Transforming Tissue Research with Open Multiplexing. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Katie White, Cell Dive Product Manager at Leica Microsystems, and Michael Smith, Cell Dive Application Manager at Leica Microsystems. I'll now pass it over to Ava to get things started.
Hello, this is Ava Amson, welcoming you to this Bite-Size Bio webinar, which today is sponsored by Leica Microsystems. Today's presentation is titled Cell Dive, Transforming Tissue Research with Open Multiplexing, and it's being presented by Katie White and Michael Smith from Leica Microsystems. Katie is the product manager for the Cell Dive Multiplexed imaging solution at Leica Microsystems. She studied mechanical engineering as an undergraduate at the University of Delaware and completed a master's degree in biomedical engineering at the University of New South Wales. Katie began working with microscopes in 2008 as a field application scientist for a high-resolution wide-field microscopy system. She has worked as a sales specialist and global product support specialist for high-resolution, super-resolution, and high-content imaging systems. Michael is the application manager for Cell Dive. He completed a Ph.D. in the genetics of DNA repair at Columbia University and a postdoctoral fellowship in genome integrity and cellular aging at Memorial Sloan Kettering Cancer Center, where he leveraged microscopy to understand the real-time dynamics of DNA homeostasis. His passion for answering biological questions using imaging technologies led him to his current position, where he provides technical expertise and customer support for the Cell Dive platform. As always, we will have a question and answer session after the presentation. So please type any questions you have into the questions box which appears on the bottom of your screen, and I'll put them to Katie and Michael at the end. The recording of the webinar will be available at bitesizebio.com. So now over to you, Michael, for the presentation.
So thank you very much for that kind introduction. So today, to give you a working example of how Cell Dive can help to transform your tissue research with multiplex imaging, I'm going to focus on a pancreatic cancer example. Pancreatic cancer is one of the highest mortality rates of any cancer. The pancreas is surrounded by a lot of highly vascularized organs, such as the duodenum, the bile duct, and pancreatic cancer often invades and metastasizes to even more distant organs like the liver, the peritoneum, the brain, the kidney, and bone. And unfortunately, conventional chemotherapy plus radiation, or in advanced diseases, chemotherapy plus targeted drug therapies, they can lengthen survival, but really they have a fairly low success rate. The 5-year survival rate for these patients is about 40%. And I think this demonstrates a lot of need for improved therapies for all pancreatic cancer patients, including new novel therapies such as immunotherapies. So we're coming to understand more and more in the current kind of scientific milieu that tumors are not monoliths. They're structurally and organizationally complex that have lots of different tissues and systems operating within them. And kind of collectively, all these tissues and systems are known as the tumor microenvironment. And it's becoming increasingly clear how essential understanding that microenvironment is for developing and using novel therapies. Different regions of the tumor, like the stroma, can behave differently from other regions in the context of the disease, and the contributions of all these different regions can underlie the often quite different responses that different patients display when treated with these emerging therapies. Beyond just the stroma, there are subregions within the tumor that can adapt to certain metabolic conditions, such as the presence or absence of strong concentrations of oxygen. So you can have regions that are both normoxic and hypoxic within the same tumor. And often these hypoxic subregions within the tumor can adapt to those conditions and actually gain dominance over the other tissues by uncontrolled cell growth. And as a result, these hypoxia adapted tissues are often more aggressive. So here, just using a small subset of biomarkers, we're going to be looking at 5 biomarkers, 2 tumor, 2 blood vessel, and 1 stroma. You can see that hypoxic regions of the tumor can be effectively classified compared to the normoxic tissue. And it's important to note here that 5 biomarkers is exceeding kind of the conventional traditional microscopy methods for analysis in the same area of the slide. And really 5 markers only scratches the surface of the information you need to understand the biological complexity of these tissues. So clearly more biomarkers are needed to characterize the cell heterogeneity in these complex tumor microenvironments. So what we wanted to do in this case is leverage the power of cell dive to look at many more markers than just 5 and probe other biological pathways in concert to see how these tumor cells might be surviving and becoming more aggressive. So we have a kind of general hypothesis to explore with this experiment, which is that the tumor cells in these hypoxic regions adapt to avoid programmed cell death and evade the cellular immune response, which would otherwise curtail their growth. So I'm going to show kind of a bird's eye view of a few different kind of biomarker panels. All of these are imaged in the same slide in the same experiment, just kind of separated so we can look at different biological pathways to show that the different phenomenon that you can look at in a single cell dive experiment. So first, we have a collection of markers to explore metabolism, such as aerobic glycolysis and the Warburg effect. And what this is going to show is the regions of the tumor that are hypoxic and normoxic. And to explore the apoptosis question, we've layered in some biomarkers such as caspase to look at which cells are undergoing apoptosis at a higher or a lower rate in the same tissue. And lastly, we're going to look at some traditional immune cell markers to explore the recruitment and differentiation and multiplication of immune cells within that tissue. So when we have all this data, the experiment becomes very complex because we have many biomarkers and many cells to explore. So what we are able to do is perform a clustering algorithm. First, we'll do some cellular phenotyping and then apply a clustering algorithm called a UMAP to this data to separate these cells into different functional clusters and then explore kind of which biomarker systems are present in each of these clusters. And so this is done by the layering of dozens of biomarkers to identify these differences that are not going to be obvious from just looking at a few biomarkers. And so in this cluster diagram, each biomarker is along the x-axis, and cells are clustered based on biomarker expression similarities across the tissue. So for example, based on the epithelial markers PCK26 and AE1 and the UMAP spatial relationships, we're going to be able to identify normal pancreas and visualize the clusters that are separate from the tumor cell clusters. So that would be numbers 4 and 6 here. And if we click again, we can see normoxic tumor tissue, and I click again. And now we see hypoxic tumor tissue. So they all cluster separately. And one of the major determinants here, we're just looking at this along the axis of GLUT1, which is a marker for those hypoxic conditions. And you can see that when GLUT1 is not present, it's not commonly present in the normoxic normal tissue, nor is it present in the normoxic tumor tissue. But in cluster 3 down at the bottom, you have a high GLUT1 expression indicating that hypoxia. So we're able to cluster these away from the other cells in the population. So where does apoptosis fall into this? So we have a couple of markers that we can look at to kind of explore the presence of apoptosis undergoing cells in this population. So we're going to look at BAC and caspase 9 as kind of 2 traditional apoptosis markers. And we can see that even among the non-tumor tissue within the pancreas, there are some cells that are low in apoptosis and some cells that are high in apoptosis. And what we find is that in the normoxic tumor tissue that expresses a low level of GLUT1, we find a higher level of apoptosis. But if we take a look at the tumor tissue that's hypoxic, what we find is a lower level of these apoptotic markers, indicating that by some mechanism, these hypoxic cells are avoiding apoptosis. And lastly, we'll look at immune cells. So we're going to use 4 different markers. We'll use CD3, which is three different markers. CD3, which is a T cell marker, as well as CD4 and CD8 to identify those subpopulations of T cells. And we can see that they cluster away from the clusters 8 and 9, cluster away from that hypoxic tumor tissue in cluster 3. So these regions are kind of immune cell deserts, and there's very limited recruitment of immune cells into these hypoxic regions, which might be another reason that they're able to survive and become more aggressive. So our finding using this panel is that cancer cells in these hypoxic regions do in fact adapt to avoid apoptosis, and they simultaneously are able to evade immune activity. So the impression I want to leave you with here is that obviously tumor cells and tumor microenvironments are incredibly complicated, and there are multiple biological pathways that are kind of at play and interacting with each other in these cells. And if we were limited to just looking at 5 or 6 biomarkers, we wouldn't be able to understand any of these interrelationships. We wouldn't understand that the hypoxic tissue also reduces recruitment of the apoptotic markers, or also, lowers the recruitment of immune cells. But through doing a multiplex imaging experiment with CellDive, we're able to see how all of these biomarkers interrelate to each other and come away with a deeper understanding of the underlying biology here. So CellDive can reveal these relationships between many different types of biological pathways that just wouldn't be clear using other imaging methods. So what is multiplex imaging? Can we zoom out and kind of understand what this kind of emerging technique is? And so I think there are a lot of definitions floating around for what multiplex imaging means, but I think the basic understanding is any type of imaging experiment that is able to look at more markers than a traditional microscopy experiment. And so that might be, traditionally 4 or 5 different markers. And so anything beyond that could be considered multiplex imaging. So we have kind of a differentiation based on the number of markers. So a lower or a more midplex experiment might look at something like 6 to 10 markers. And this might be accomplished through something like spectral unmixing. Many different fluorophores are present in the sample, and they're able to use software and optics to separate those different spectra and then visualize the different markers. If you want to go beyond just 6 to 10 markers and look into many more, like we've done in this experiment here, it's important to use something like an iterative hyperplex system. And so what this does is it uses multiple rounds of staining and either bleaching or dye inactivation or stripping of antibodies to iteratively probe and revisualize the sample. And you can see in these images at the bottom that multiple biomarkers are being layered on top of each other with each round. And this really allows you to get past the kind of traditional limitations of these platforms and look at many more markers, 10, 20, 30, 40, 50, 60, 100. And CellDive is part of this family of technologies. So there are a lot of considerations, and I'm sure people who are considering this space probably have a lot of questions about how to get started. And there are a lot of questions that can go through the mind of the researcher, such as, can I get started quickly? These seem like complicated experiments. Is it going to be something I can pick up, or is it going to require lead times to bring in the technology that are kind of unacceptable in a fast-moving research space? Can I design studies to answer my unique question? Is this going to be flexible enough to be able to be applicable to what I'm studying? Researchers are looking at very specific issues and specific parts of tumors. Is this going to work for my unique system? And am I going to be able to adapt this study? If my research needs change or grow over time, am I going to be able to be flexible with this? Or am I going to be locked into some kind of kit or method that might not work throughout the lifetime of my research? Can I return to my slides for downstream processing or more imaging? I'm sure we've all had experiments that we wish we could have gone back to and just looked at 1 or 2 more things. Some of these methods are destructive, and especially with precious or expensive tissues, that can be a real challenge. So am I going to be able to go back and have the flexibility to add in a few markers if my research question evolves during the study? Is the workflow reliable? Is it going to be something that produces the same kind of results every time on similar tissues, or is it going to produce swings and batch effects that are going to make analysis more challenging? Can I trust the data? Is this going to be replicable from the beginning of one study to the end of a study, which could be months or years later? Is it going to be replicable in a different lab? Is it going to be replicable many years later? Can I trust the data? And am I going to be able to scale up if I need to? If I start an experiment that looks at 10 slides of tissue, but I decide I want to move up to 100 or 1,000, is this technology going to be able to grow with me and my needs? And lastly, am I going to get the high quality image data I need for robust analysis? Of course, everything that's extracted from imaging data sets depends on the quality of those images. And if these image quality isn't good enough, then we can get incorrect or unreliable analysis from that. So am I going to have the image quality to actually get results I can trust? So I'm going to kick it over to Katie White, our product manager here at CellDive, to tell you more about how CellDive can answer and help you with some of those important questions and bring the world of multiplexing to you.
Thank you, Michael, for a great introduction to the power of multiplexing. And in this next section, I'd like to tell you a little bit more about CellDive specifically. So what is CellDive? At first glance, you might assume it's an automated imager, but really it's a complete multiplexed imaging solution. And this graphic here shows the components of the CellDive solution. The first component is the validated antibody list. And this is a list of over 350 rigorously validated antibodies that have been proven to work with the CellDive process. And doing this type of validation takes considerable time and money. So access to this list is a huge productivity jumpstart. Second, we have the patented workflow for CellDive. And this iterative staining and dye inactivation workflow is what allows us to stain and image a sample repeatedly with new biomarkers in each round. Next up is the acquisition software. And built into this software is a huge amount of technology around image calibration and correction. And these calibrations and corrections ensures that these large ROIs that we collect for CellDive can be seamlessly stitched and precisely aligned to enable downstream single cell analysis. The next is the CellDive imager, and it is a very robust automated microscope with a number of customizations to make it a dedicated tissue imager, including an onboard barcode reader, 5-channel capability, and compatibility with automation systems for sample loading. The final component is the analysis software. And in this space, we've partnered with Indica Labs and authored their tissue pathology software called Halo. And this software is very well known in the tissue analysis space for balancing power and ease of use really well. So on that last slide, I talked through the components of the CellDive solution, but that doesn't really explain how CellDive works. And what you see here is a high-level overview of the CellDive workflow. We begin our process with your tissue sample. And the first thing that we do is we prepare those samples using a well-defined slide preparation process that it includes a patented antigen retrieval technique, but also more standard processes like dewaxing, clearing, etc. So after the slides are prepared, we put them on the imager and we use quick low magnification scans to define the region or regions that will be imaged for the study. When that's complete, we get into the iterative part of the study, the imaging rounds. And first, we stain with up to 4 dye-conjugated antibodies. We perform our biomarker imaging, and then we inactivate the dyes or turn them off with our chemical dye inactivation solution. So this process doesn't remove antibodies or photobleach the sample, so it's very gentle and tissue preserving. And this is the step that allows us to stain over and over with new antibodies. When that's complete, we image our autofluorescence signal again, and these images are used for computational autofluorescence removal, and then that concludes an imaging round. So those 4 steps are repeated for different biomarkers until the study is complete. From there, images are used as input to image analysis programs to do comprehensive segmentation, classification, and spatial analysis of the sample to answer research questions. So in this section, I'd like to explore in a bit more detail what we really mean when we say that CellDive is an open multiplexing system. So we know that getting started with multiplexing can be overwhelming, especially when it comes to building panels. And to help you get started with confidence, CellDive comes with an extensive list of 350 antibodies that have been rigorously characterized to ensure they will work with the CellDive process. And you can leverage that and build your panel entirely with antibodies from this list, or you can use the proven CellDive characterization process to validate your own antibodies. Additionally, being an open multiplexing system means that we're agnostic to the method you choose for multiplexing. So if you'd like to use a third-party multiplexing kit, or you use one of the open source multiplexing methods, you can do that with CellDive. It's also important to mention that we don't dictate where you source your antibodies or reagents. So you're free to use the suppliers that you like and that you trust, while simultaneously keeping your costs down. Finally, I think the real power comes from the fact that you can combine any or all of these strategies to build your completely custom panel. For example, you can begin with an 8-marker multiplexing kit that covers maybe some core immune cell markers, supplement with a number of markers from the CellDive validated antibody list, and then add in a few of your favorite antibodies that you've validated yourself, and voila, you have a biomarker panel that is completely customized to your specific research question. With CellDive, you're also free to choose what you'd like to image. So depending on what you're interested in, you can decide to image your whole tissue and select a single area up to 20 by 45 millimeters in size, which is huge. Or you could identify smaller regions of interest to examine specific structures. Imaging areas aren't constrained to any size or shape either, so you can image exactly what you need, but only what you need. CellDive protocols are also incredibly comprehensive as well. So they detail all steps of the multiplexing workflow, including benchwork steps, QC steps, and of course, imaging steps. And this ensures that the right processes happen at the right time for the right samples. But even with the most well-designed protocol, you might need to pivot or change something while the study is in process, and that's possible with CellDive. So maybe early examination of your data gives you a new avenue to explore. It's not an issue. You can add a couple more rounds of imaging at the end of your study. Or maybe a great new biomarker of interest becomes available a year after you complete a study. And if you saved the slides, you can pull them out of the fridge and you can stain them again. With CellDive, it's never too late to adapt your study design to respond to your research questions. So scalability and efficiency is important to everyone, not just as it pertains to multiplexing, but to scientific research generally. So how can CellDive help you become more efficient and help you scale your work? So when you think about scalability, if you're like me, you immediately picture sort of hardware for automation. But without a framework and software to enable that automation, it kind of doesn't work. The automation hardware needs to know what to do with your samples. And this tracking becomes even more complex with multiplexing, where you may have many samples or even studies running concurrently, and somehow you need to keep track of what to do next with each and every slide. And that framework or backbone of multiplex study management was one of the driving forces behind the development of the CellDive acquisition software. So with input from an integrated barcode reader in the imager, the software automatically tracks slides through a multiplexing study and performs exactly the right action for that slide at exactly the right time. In addition to tracking multiple slides, the software can handle multiple studies at a time, making it perfect for a multi-user environment or just a busy lab balancing multiple studies or cohorts at the same time. The software also plays a big role in compatibility with automation. Once a protocol is defined and regions are selected for all slides, there is no user interaction required in subsequent imaging steps. The slide is loaded, the barcode is read, and the system knows exactly what to do according to the protocol. And this allows for easy integration with automation solutions. Another aspect of this CellDive workflow that allows for scalability is how slides can be processed in batches. And to explore this in more detail, let's examine this graphic of the iterative portion of the CellDive workflow. And in this workflow, we have 2 benchwork steps where we actually manipulate the sample, and those are the biomarker staining and dye inactivation steps. And we have 2 imaging steps where the slides are imaged. And with CellDive, the benchwork steps happen off the imager, which allows for batch processing of slides through those steps. So if we take a closer look at what that manual workflow would look like for maybe 3 slides, this is kind of what we would see. So each of the scientist icons around the outside of this cycle represents a touch point in the cycle, and its size roughly represents the duration of that touch point. So if we start from the top left at biomarker staining, which is a benchwork step, you can see that there are a couple touch points here that are kind of longish in duration. And as we move into biomarker imaging, we have 3 touch points. So in this example, we have 1 for each slide that we're processing, but they're very short, just the amount of time to load each slide into the imager and push go. And that pattern is repeated on the bottom half of the cycle for dye inactivation. A couple longer touch points to perform dye inactivation on all slides at once, and then short touch points to load the slides into the imager one by one. And this batch processing workflow is advantageous for a few very important reasons. So number 1, since all slides in a study can move through the benchwork steps together, you can have confidence that they've been treated as similarly as possible. You'll be staining with the same antibody solution on the same day. The environmental conditions will be the same. The incubation times will be the same, etc. The workflow also enables you to scale your research, even if you don't have the means to automate the process. So whether you're working on 3 slides or 10, the benchwork portion of this workflow doesn't change much. The touch points get slightly longer to address more samples, but it certainly doesn't scale linearly with the number of slides. And finally, the fact that benchwork steps happen off the imager allows you to keep that imager working as much as possible. It doesn't have to sit around while slides are being stained or going through dye inactivation. That time can be used for other slides, other users, and other studies. So what about automation? In my years of talking with researchers in this space, one thing is a recurring theme. Everyone wants automation in multiplexing, but not everyone wants the same automation. So from those who would like to just set it and forget it for 2 to 3 slides, to those that need complete staining and imaging automation for cohorts of 50 or more slides, one thing is very clear. A one-size-fits-all approach to automation just doesn't work. And this is why I think it's so important in this space to enable automation, but allow you to dictate what that automation looks like based upon the throughput you require, your budget, or your access to existing equipment. So for CellDive, automation is enabled by the Clickwell cover slip free slide holder. And Clickwell is unique because it allows you to perform all of your benchwork and imaging steps right in the Clickwell, or allows you to repeatedly remove and reinstall slides from the Clickwell for processing elsewhere. So what are some of the ways that the CellDive workflow can be automated? So in looking at this manual workflow, the thing that jumps out to me first, especially if you'll be imaging a larger number of slides, would be to eliminate the touch points in each of the imaging quadrants. And Clickwell was designed with the footprint of a standard multi-well plate, so nearly any robotic arm can be integrated into the system to automatically load plates into and out of the imager. And with that type of automation, the touch points for biomarker labeling and dye inactivation, our benchwork steps, they wouldn't change. But we've already discussed that even with a larger number of slides, those steps can still be done for all slides in a batch fashion. And after the benchwork steps are complete, there would be one touch point in the beginning of the imaging segment to load the slides into the input stack for the robotic arm and initiate the imaging process. From there, slides would be loaded into the imager and imaged one by one until all slides have been imaged. And that one automation step increases the amount of unintended time really significantly. So that means that for the entire imaging time for all slides, you're free to perform other tasks. It can also enable overnight imaging to allow you to take advantage of every hour of every day. We also hear from researchers that they would like to automate the benchwork parts of this process, and that's possible with Clickwell as well. So the repeatable slide positioning and ability to easily install and remove slides from Clickwell allows for the use of commercial auto stainers to automate staining and dye inactivation. And this diagram shows what that workflow would look like when you combine the robotic arm with a commercial auto stainer to automate the benchwork parts of the process. Similar to the manual workflow, the slides would be processed in the auto stainer in a batch fashion. As for touch points, in this type of workflow, there are just 4 at each of the transitions between the auto stainer and robotic arm, or vice versa. And again, this expands the unattended time between touch points, even with very, very large number of slides. The last thing about this open approach is that it also enables your level of automation to scale with your research. So maybe today a manual workflow is fine, but in a year you get a huge grant for a major study. And with CellDive, you can do that type of scale up just by adding supporting equipment, not additional imagers. So we do have one more automation option to make this process even a little bit more hands-off. And this is a new solution that we've developed exclusively for CellDive with Advanced Solutions Life Sciences. It's a customization of their BioAssemblybot 200 platform to completely automate a full half round of staining and imaging for CellDive. And the cool thing about this solution is that it's a robotic arm with interchangeable attachments. In this case, an automated pipetter and a plate gripper that allows us to automate both staining and imaging or dye inactivation and imaging to further expand the unattended time. And with this solution, there are only 2 touch points per round. So you load up to 15 slides, you set up the system with required reagents and tips, and you push go. On this slide, we see the BioAssembly bot working on a set of slides in ClickWell slide holders. In this scenario, benchwork steps are done directly in the ClickWell by pipetting reagents in and out. So we can do the entire staining or dye inactivation process right in the ClickWell, and we can do it for up to 15 slides at a time. BAB also comes with pre-configured bio apps that cover all the main workflows for CellDive. So that includes staining, dye inactivation, and even an image-only mode that allows BAB to just act as a simple plate loader. So there's nearly no overhead to getting started. And as you can see here, BAB and CellDive are integrated seamlessly. So BAB performs all the steps for staining or dye inactivation, then can load samples directly into the CellDive imager, and the imager does the rest. It will carry out the right imaging actions based upon the slide's barcode and the protocol. So essentially, you load the slides, and when you come back, everything is done. You just get to review final stitched images for quality control. Finally, these custom workflows were designed specifically to keep the imager busy. So anytime there is a slide that's finished imaging, there is another slide right behind it that's ready to start. Last but certainly not least, how do you know you can trust the data you get from CellDive? Multiplexing has exploded in popularity in recent years, and it seems like every day there's a new system or new product in this space. And Leica Microsystems might not be the first company you think about when you think about spatial biology, but CellDive and the technology that it's based upon has a long history in this space. The CellDive platform and the workflow has been in development for over a decade. In fact, the first paper was published way back in 2008. And since then, countless hours from both the developers of the technology as well as prominent early adopters worldwide have gone into testing, refining, and validating the entire CellDive workflow. There have also been over 40 peer-reviewed publications that leverage CellDive in recent years. And finally, as a real-life application for this type of technology, CellDive has been used in biopharma since 2015 as a robust way to stratify patient populations for clinical trials. So this iterative staining workflow is the backbone of the CellDive process. So it would be the backbone of your research as well. For something so important, it's imperative that it just works, and it works the first time, and it works every time. And the years of research that have used this process guarantee just that. So you can get started with confidence in that workflow. Over the years, collaborators have run some huge studies with this process so that we know it works for 60+ biomarkers. The largest study that was done used over 120 biomarkers with this process. It's also proven to be very gentle. So at the end of your studies, your tissue is still intact and can be used for downstream processing to be stored or to revisit in the future. And finally, our patented 2-step antigen retrieval has been shown to provide better antibody binding and increased staining intensity for better quality data. Next up is the CellDive imager. And this is what Leica does. This is what we're known for, making high quality microscope systems. And the CellDive imager is no exception. This purpose-built, fully automated tissue imaging system was engineered for precision, speed, and sensitivity to make sure our images match the quality you expect from Leica, but more importantly, so that you get robust downstream analysis results. We can also capture nearly the entire imageable area of the microscope slide, so you aren't limited in the size of the tissue you would like to examine. And finally, we all know the pain of imaging a very, very large sample and then having just a few out of focus frames to ruin it. And the cell dive imager takes advantage of advanced slide mapping and multiple autofocus routines to ensure every frame is in focus every time. So I'm a microscopist. That's sort of my background. So I love to look at beautiful image data. But the truth of the matter is that the images that we get are really a means to an end. It's the data, the analysis results that we get downstream that we use to generate insights. And while that's absolutely true, I don't think we can minimize the importance of good image data to start with, because even the most advanced analysis pipelines can't fix poor images. It's that whole garbage in, garbage out idea. And the cell dive software takes the guesswork and the effort out of getting to your final image. Our custom calibration plate and automatic calibration routine was developed over 10 years to produce seamlessly stitched and precisely aligned multiplex data. And the images here are a great example. So what we see here is subnuclear registration structures from different rounds. We have Ki67 from round 1 of imaging and DAPI from round 3. And even those were imaged at 2 totally different rounds, we get that perfect registration between the channels. And the best thing about the whole process, the calibration and correction process, is that it's completely automatic. You run the automatic calibration routine once a month, and those calibrations are automatically applied to your images such that all you see is the final product, a corrected, stitched, and aligned data set. These calibrations also enable correlation across multiple samples, multiple studies, and even multiple imagers. So to summarize, Michael showed us a great example of how multiplexed imaging can answer complex questions that can't be fully explored with other imaging methods. We also highlighted how open multiplexing with CellDive lets your research dictate how you run your multiplexing study, including how to build your antibody panel, which antibodies to use, and even the level of automation that's needed. Finally, I hope we've shown that regardless of how you choose to proceed with your study, the data quality you get from CellDive doesn't change. You'll always get crystal clear whole tissue images that enable you to deepen your understanding of the tissue microenvironment through outstanding spatial mapping of single cells. Thank you all for joining us today. We appreciate your time. We can turn it over for some questions.
Thank you, Michael and Katie, for your informative presentation. Before we proceed with our Q&A session, we ask that you participate in a series of poll questions that will be displayed on your screen. So we'll go ahead and proceed with those. Our first question here, do you currently use multiplexed imaging in your research? Please select one of these answers on your screen. Again, the question is, do you currently use multiplexed imaging in your research? All right, we'll proceed to the next question. What method do you currently use to analyze tissue selections? Please select all that apply. Again, what method do you currently use to analyze tissue selections? And our last question here, are you interested in learning more about CellDive? Please select your answer. Great, thank you so much. All right, we've got Katie and Michael here. Wonderful. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. All right, our first question here is for Michael. Michael, how long does it take to validate an antibody, and how much does it cost?
That's a great question. If you're looking to bring in an antibody that's not currently on the cell dive validated antibodies list, we provide instructions for a pretty exhaustive 3-step process for validating antibodies. And we estimate that it would take around 32 hours of labor and about $1,500 to $3,000 in reagent costs to go through that process on a per antibody basis.
Great, thank you. Next question here is also for Michael. What type of image correction does CellDive perform?
So the CellDive imager comes with a specialized calibration plate that's loaded into the imager and is used to perform a variety of corrections that ensure high imaging quality without a lot of intervention and corrections done manually by the user. And there are, I believe, over 10 different corrections that go into the system. But just to list a few of those, the objectives are centered. There are blank glass artifacts that are subtracted. We perform flat field corrections, intensity calibrations, distortion corrections, stage alignment, and of course, image alignment and registration. And autofluorescence is removed from each image based on an autofluorescence image, not off the calibration plate. So there are a variety of different routine imaging calibrations that are performed without user intervention.
Great, thank you. Katie, this next question is for you. To do multiplexing at scale, do you have to automate?
That's a great question. And the answer there is no. So we have customers that are doing multiplexing from at scale and using either a fully automated solution or not automating at all, doing everything manually. And as we kind of mentioned in the webinar, to do the cell dive process manually, whether you're doing 2 slides or 10 slides, the workflow really doesn't change all that much, especially with the bench work. You know, a lot of the time in bench work processes is spent, getting your reagents, mixing up the solutions that you need to use. And whether you're doing 2 slides or 10 slides, that only needs to be done once. And then you just need to sort of either pipette those reagents onto 2 slides or 10 slides, or, dip them into a staining dish. So we do have customers that run studies with, 10, 20, 30 slides, and they do it all completely manually. So it is possible.
Great, thank you. Another question for you, Katie. The advertising says there is a list of validated conjugated primary antibodies for use in CellDive. Where do I find this list?
So that list is actually part of the solution. So when you purchase a system, that list comes along with the system for you to use to design your study. So it is not something that we give away. There is a lot of value in the list. So as Michael mentioned, to validate each of those antibodies, it takes 32 hours of time and $1,500 to $3,000. So that is something that we sell as a part of the solution.
Great, thank you. And Katie, another question for you here. Can the imager do thicker sections, 30 microns, 50 microns?
That's a great one. So the short answer there is no. So CellDive is an inherently 2D imaging solution. So we advise our customers that tissue sections around 3 to 5 microns thick is where we need to be. And the one thing to consider when you're thinking about doing multiplexing on thicker samples is the volume of data. So if you are doing whole tissue imaging and you're doing it through 30 microns or 50 microns, just the avalanche of data that is produced, and the difficulties that would be there for downstream analysis cannot be underestimated. But short answer, CellDive is a 2D tissue imager, 2D multiplexing system.
Great, thank you. Well, we will go ahead and wrap up there. Michael, Katie, do you have any final comments for our audience?
No, I don't think so.
Thank you so much for your attention.
Wonderful. Well, thank you again, Michael and Katie, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.